just rounding up a few people who are really absorbed in the posters and the displays. Good to see that you're all visiting them. And I'm just about to introduce you to our afternoon session, which is going to be chaired by Tim Wardlaw. So Tim, would you like to come up and take over the session? Thank you.
Right. Welcome back. I feel like I'm finally in Brisbane. So session 3 this afternoon is land management for sustainable ecosystem services. And Mark's keeping the time. You keeping the time? You the timekeeper? He's going to be elbowing me when there's 3 minutes to go and kicking me when there's 1 minute to go and hitting me over the head with a mallet when time's up. So I'll pass that on to you. I haven't got the speakers, so I go to the next session. Maree's going to do this. There we are. So Maree, do I need to introduce myself? It's in program anyway. I'm the principal investigator for WARA, and I'm a passionate TERN supporter, which is why I'm here. But let's start with Maree.
Thanks very much, Tim. Well, my name is Maree Bowen. I'm with the Queensland Department of Agriculture and Fisheries in the Extensive Livestock Systems Group, and I'll be talking to you today about the tension between achieving profitable grazing systems while maintaining land condition and sustainability. I'd like to start by first acknowledging my key colleague and co-author on this work, Fred Chudley. Fred's now retired, but brought the economist skills and expertise to our project. Our work was funded by the Queensland Drought and Climate Adaptation Program and also the Australian Government Future Drought Fund. So Queensland's grazing lands occupy more than 80% of the state. And so the decisions made by our land managers, our primary producers, are very important in terms of consequences for the environment. So what is this tension that I talk about for Queensland's grazing lands? Well, we have good empirical evidence that supports sustainable grazing management practices. We know that safe stocking rates based on safe pasture utilisation rates will maintain land condition. And we have evidence that under some circumstances a reduction in stocking rate combined with wet season spelling can improve land condition. And based on these principles, there's been a concerted extension effort over more than 3 decades to promote sustainable grazing management practices to primary producers, to our grazing managers. But in spite of this, we have evidence of ongoing application of higher stocking rates broadly across the landscape, higher than recommended to maintain land condition. And we have evidence, good evidence for continuing land condition decline across our grazing lands. And that comes from on-ground data and satellite imagery. So to understand what's going on here, we need to look first at try and understand our perennial grass pasture systems in Queensland. So we know that our perennial grass pasture systems are quite resilient to heavy grazing. We've got a number of long-term grazing research trials that have run throughout Queensland, and we know from these sites that it can take decades to seriously affect the land condition at high pasture utilisation rates. And that means that the cost to the primary producer of that degraded land is much diluted due to the time value of money. Furthermore, we've got evidence that these perennial grass pastures can be quite slow to recover once grazing pressure is reduced. And we've got a number of research sites indicating that With regular wet season spelling at one site or in one trial, there was no impact on land condition over 13 years of regular wet season spelling. That site was in North Queensland. At two other research sites in Central and North Queensland, there was no impact on land condition over 3 or 5 years respectively from regular wet season spelling. So this is problematic in terms of the decision making for producers. And there was an additional site in the Burdekin actually, which ran over 10 years. This site implemented reduced stocking rates and regular wet season spelling, and the authors concluded that it would take more than 10 years of this quite serious grazing management intervention in order to meet current targets for water quality. So these are the issues we're facing in terms of practice change. And an overlay to all this is the substantial climate variability that you'd all be well aware of. And this means that any changes in land condition due to grazing management rather than due to just the year-to-year climate variability can be very difficult to discern for the grazing manager. They're not sure whether a change has occurred due to their own management or whether it's just climate variability. Now, it's also important to look at the characteristics of our extensive beef production systems. The majority of our graziers are beef producers at the present time, and it's important to understand those systems and what's driving the management decisions. So broadly, we know that beef producers are under some financial pressure, and this is because there's a disconnect between asset values, so their land value, and the returns that can be made from that land. Broadly, across the sector, there are high debt levels and also a declining trend in terms of trade. We've also got data to indicate that grazing managers who are already in financial difficulty or who have lower levels of equity are likely to apply heavy stocking rates because they believe they will make more money, and we have survey data to indicate that. So it's evident or apparent that to understand this decision making by primary producers, we really need some sound economic analysis of grazing management strategies and the consequences of these decisions. And our perspective in the department is to understand what's driving the decisions of primary producers or land managers, and also to provide better information and a framework to enable better decision making. So we believe it's important to conduct a sound economic analysis, but it must be a valid economic analysis. And there are two components to a valid economic analysis of grazing management strategies. The first is that it is critical that the correct economic method is applied in this type of analysis. And by this we mean a marginal economic analysis. And so that means we're modelling a change in grazing management. So we alter over time the herd performance, the performance of the cattle herd, and the inputs of our base or starting scenario to construct new alternative scenarios. And this type of analysis provides the information that a grazing manager requires to make an informed decision about a change to their current grazing management strategy. An analysis that doesn't include this transition from an existing strategy to an alternative, it actually ignores most of the critical financial and risk management aspects of that decision for the producer. And these components act as the greatest impediment to change. And unfortunately, they have been ignored in many analyses of the impact of stocking rates. So the second component of a valid economic analysis, we need the correct method, but it's also critical to have the correct perspective. And I was talking before about the long-term research, grazing research trials we've had throughout the state. And these have lots of excellent data, but in order to obtain relevant insights for industry, we must interpret those results and apply them in a commercially relevant context. Otherwise, they're just not relevant to industry. So by doing this, we need to apply industry costs, and we need to adjust the management strategies so that they resemble industry practice and accepted performance. We also need to extrapolate the modelling from small paddock scenarios. So often these experiments are run in very small paddocks that aren't much smaller than what you'd expect commercially, and they're run with castrated male animals, so steers or wethers. So we need to extrapolate to the breeder herd, a herd of breeding females, and a scenario that represents a commercial reality for producers. And there's actually there's a real challenge in doing that, because we have a critical lack of information to support converting a change in steer performance or wether sheep performance, to changes in breeding herd performance for a commercial beef property or sheep property. So it's actually quite challenging to do that appropriately. So when you look across the literature, unfortunately, there are very few studies that do report a marginal analysis, which is a sound and appropriate methodology. So a marginal analysis of grazing management strategies, either at the paddock level or the whole property level. And this has also been identified by others in previous reviews. So Moravec et al. And Denny and Moravec have identified this problem as well. Of those papers that do report a marginal analysis of change, there are only 4. The first of these reports application of data from a grazing experiment with steers in central Queensland, and that's the Galloway Plains trial, for those who may know that. But this analysis was an appropriate marginal analysis of change to look at the economic consequences. And there are 3 additional papers that report property-level economic modelling and appropriate analysis of change. 2 of these are based on central Queensland environment, and the fourth, or the third of those series, is based on the Texas rangeland environment. But the key point I want to make is that all 4 of those analyses, which were conducted using an appropriate economic method and an appropriate perspective, were all consistent in their conclusion that there is a very strong economic incentive for primary producers to apply high pasture utilisation rates, even when this results in declining land condition over time and therefore declining individual animal performance. Another analysis providing useful insights about land condition decline includes this paper that was actually published in 91 by Passmore and Brown. It's a stochastic analysis, and interestingly, this paper determined that the economically optimal stocking rate for grazing land managers rises with lower property sizes, higher discount rates, higher commodity prices, and declining risk aversion. And they concluded that a strategy of high stocking rates with potential for land degradation is an optimal response to the economic and social factors that confront graziers. And just for those who aren't familiar with some of these economic terms, high discount rates means a preference for cash flow or profits now in the near term, and both high discount rates and declining or low risk aversion, both declining risk aversion means that you want to profit, or you tend to profit maximise, or you're looking to profit maximise, but both of those preferences are influenced by debt and financial pressure. So debt, financial pressure, we spoke about that being an issue broadly in the beef industry, small property sizes, these are all contributing to these high stocking rates that we're seeing across the industry. Now, with the high commodity prices, so that means high beef prices or high wool prices if you're talking about sheep. Now, it might seem somewhat counterintuitive that high commodity prices can also lead to high stocking rates, but there are several other papers that have also indicated this same effect. And the reason for that is because of what economists call the opportunity cost. So there's an opportunity cost to the producers of not taking advantage of those high prices while they're there. Because one thing's for sure, in the agricultural industry, they won't be there for long, so people make hay while the sun shines. So that brings me to my conclusions. And to sum up, those analyses which have been conducted using sound economic analyses indicate that there is a strong financial incentive to apply high pasture utilisation rates, even when this results in declining land condition and individual animal performance. So this must be recognised and understood before we can find solutions to address land degradation. And importantly, to develop solutions that are economically viable for producers and economically attractive for them to adopt, it is critical to apply a marginal economic analysis at the property and herd level to really understand the impacts and the consequences for their business. And such an analysis will appropriately consider the challenges in transitioning from one management strategy to another, so implementing a strategy that we believe will be more sustainable, for instance. So that brings me to the end. Thank you very much.
Hey folks, thank you for having me. My name is Will Edwards, and although this is a session that identifies sort of ecosystem services, what I'm going to be talking about is something slightly different. It's not a direct ecosystem service. It's actually an ecosystem function and an opportunity I think we have to develop a method to rapidly assess an ecosystem function that's vitally important. Now, most of you will understand that what we currently know, or what we currently do in terms of monitoring what we could call biodiversity, is centered on big things. You know, it's acoustic recordings or ultrasonic recordings or camera traps. And they monitor big things, and they're used to provide estimates of sort of the presence of something or the loss of something from somewhere, or are combined to some sort of community assemblage or community metric. What I want to talk about is something that's slightly different than that. It's resolvable very quickly and over a very small amount of time, so I think we have the opportunity to generate point estimates of this process. And for most of you, I don't know, I mean, I think most of you probably know, one of the principal ways that plants can have their offspring avoid the effects of fire is to engage ants in taking them and moving them underground. And so many plants produce seeds with a fatty, rich rhizome appendage. The ant takes it, carries it to the ground, into its nest, cuts it off, feeds it to its larvae, the appendage to its larvae, and some of them get stuck, or some of them get lost underground in the nest. And that's a good way of avoiding fire. That mechanism, and there are many different types of it, has evolved and is prevalent in almost every place in the country where there is fire, and that is almost every place in the country. Here is a figure that shows the relationship between the amount that a plant is invested in enticing that relationship and the weight of the seed. And it's for the orange ones are for acacia species, and the blue ones are for a whole range of other species in many different genera and many different families. No matter how you analyze that in a phylogenetic context, if you take the family means and produce that figure, you'll get the same slope. If you take the genus means independent of their family influence, you will get the same slope. Those things, that consistency, implies that that relationship has been maintained and selected for incredibly long periods in the evolution of Australian plants. The alarming news, I guess, this is an excellent study, and I don't know, maybe some people in the audience here actually were involved in this. It's a study from South Australia where the authors looked at the composition of both ants and plants in co-occurring locations and tried to work out what it was that structured them and projected that into the future to see whether or not the plants and animals would move in the same way. And their overall conclusion was that there was going to be, we might expect a relatively high degree of community reorganisation and change in ecosystem function because those two things are going to be changing differently. So, I think it's a probably good idea that we try and actually have an understanding of what that function might be. And what I base this thing on is a thing called rapid ecosystem function assessment. There are sort of 4 principles to rapid ecosystem function assessment. It should be low tech, it should be easy to use, repeatable, and really cost efficient. And now, because my conquer the world sort of ambition is to entice a lot of people to do this, not just necessarily the ecosystem processor sites of TERN, because this process occurs across all of Australia. It should be a thing that we can ask people to do that doesn't impose any sort of burden on them. They could just do this relatively easy. And so without producing too much of a burden. And so I have generated, and I have, strangely enough, a show and tell moment. A small sensor that looks like this. And it's just essentially a plate that has a position for a number of seeds, a small camera at the top that focuses on that space. It's Bluetooth enabled, so you can generate an app which you can just get the data from. That data can then be, the images that it takes, there's a software that we've written that converts that to just a simple CSV file of the time and the number that are remaining. And you can put that through a program or like a simple analysis pathway to produce a result. It's a simple thing. Do it in the morning. Go and do the work that you're really going to your field site to do. Pick this up in the end. Turn it off. We will do the rest. Better still, we could do all the rest because most of it, once you've uploaded the data to the app, can be just sent to a central repository, and it can come along with a whole load of different metadata that might be associated with it, the time of the year, the absolute location, what type of plants were, or what type of seeds you used, and so on and so on. So, yeah, that's my plan to rule the world, I guess. Didn't know that was coming, did you? I guess I don't want to leave you with a sad story or a frightening story. If we go back to those two things, the fact that that relationship and the selective pressure for that relationship lasted so long and went and persisted across that incredibly long evolutionary time means that probably the community composition of ants and plants that are projected to be different into the future were also probably different in the past. And so that relationship still persisted even though there was not necessarily the same associations that we see today. There may well be some level of functional redundancy. It doesn't really matter who is there. They're all going to do the same sort of thing. But I guess we don't want to actually trust our future into some strange deduction that I've just made on the stage here. The big question here, I guess you're all interested in is, well, how well does it work? And the unfortunate answer is I don't really know, but I hope to know by the end of the year. Currently, my third-year ecology class is trialling it, so how many would they put out in a particular area and what sort of configuration? Should we use natural seeds? Can we use a proxy, like a standard seed, like black rice or something, cut in half? I have an IoT student who's helping me, I hope, to generate the app that will download the data to your device. And we're intending to implement this on the TERN WA Transacts at the end of this year. Finally, there's probably a whole range of other different contexts you could use this device in. In far north Queensland, where I live, we have invasive yellow crazy ants, and rather than assess what they've done to the other types of ants, you could potentially use this as a mechanism to see whether or not the functioning of the community of ants has changed with the introduction of the yellow crazy ant. So, thank you very much.
Chat to you today. So I'm going to talk to you today about a collaboration that we did between Curtin University and researchers at the Department of Water and Environmental Management. And we were looking at whether or not satellite-derived products on land surface water usage, measured as evapotranspiration, and marine quality products can be used to identify the land use and land cover sources of threats to the health and water quality within estuarine ecosystems. So in particular, we were looking at whether or not the impacts of activities that happen within the estuary catchment systems, such as increasing urbanisation, increasing agriculture, increasing use of fertilisers, irrigation, and how these impacts affect the water quality within the estuaries, and whether or not we can tell that by using evapotranspiration products from a satellite. So we also wanted to use our mapping products to actually provide useful products for the department around their estuarine management monitoring programs that they do at these sites. So we produce maps of soil and vegetation water usage, chlorophyll A concentrations, and total suspended matter concentrations. So for our methods, we use the Sentinel-2 and Sentinel-3 satellites for our coastal water quality products, but I'll just present to you the Sentinel-2 products here. So what we did, and this work on the marine products was done by my co-author, Professor David Antoine at Curtin University. So it's deriving ocean colour, and from that you can estimate parameters such as the total suspended matter and the chlorophyll concentration and other things such as CDOM. The accuracy of these parameters is going to be dependent on a number of factors such as the atmospheric corrections to the data, the model that you've used, the depth of the water that you're looking at, which came into play in some of our sites. So we use the acolyte algorithms that are referenced here. We did the monthly median over our chosen month to compare with the land surface water use products, and our mapping products are at a resolution of 10 metres per pixel. So for our land surface water use products, the parameter that we're looking at that we've derived is the actual physical parameter of evapotranspiration. So that's the total of evaporation and transpiration. So it's a total loss of water from the surface through those combined processes in terms of the units of millimetres per day per pixel in the mapping product. And again, we're at the resolution of 10 metres per pixel. We're using Sentinel-2. So the model that was used is referenced here, but it's a new version, but based on this model here. It's something that I've been working on for a number of years, but it's based on multispectral data, vegetation indices, and it incorporates climate data as well through the format of potential evapotranspiration and rainfall, daily rainfall products. So our 3 estuary sites are distributed, as you can see here. We've got the Matuwara, Kuiniwadu, and the Mirich estuaries. So these 3 sites are very different in terms of their climate zone, in terms of the amount of rainfall that they receive and when they receive it, so when their wet and dry season is throughout the year, and also in terms of the size of their catchment. So you can see that the northern 2 are very large, whereas Oyster Harbour just fits within one sentinel image, so it's very different. So here's just a view of those 3 sites. So our Fitzroy site is really our kind of pristine end member site. In terms of land use and land cover within the estuary, the main land use is really grazing native vegetation. Beyond that, so a bit of dry land agriculture, but beyond that it is predominantly national park and residual native cover. The same is predominantly true in the Gascoyne. The main land use again is grazing native vegetation, with the exception of a small area around Carnarvon, and there you have a lot of irrigated cropping and in particular irrigated stone fruit and other agriculture. And the Oyster Harbour is really our other end member site in terms of having the most urbanisation and the most intensive agricultural practices. So you have a range of different types of plantation, different types of grazing, and you also have cropping production from different forest and agriculture. So if we look now at our evapotranspiration mosaic, so each pixel here represents millimetres per day in water loss out of the surface from the combined processes of soil water loss, evaporation, and evaporation from vegetation canopy cover, and transpiration from vegetation moving water out of the soil. So you can see here the wet season mosaic for the Fitzroy, and here the dry season, noticeably different for the Fitzroy as well. Because it is dominated by rainfall dependent vegetation. So here's just a zoom in so you can see a little bit of the level of detail. There's a bit of topography here in this image. So here's the dry season and here's the wet season, so you can really see those runoff zones where you have those rainfall fed gullies. Here is something that's just an anomaly really to see in this kind of area. You could probably guess what it is from the shape. So that's it in the dry season, that's it in the wet season. Amazing to see in such an arid environment. This is irrigated cropping up there in the Fitzroy. So now if we look at the Gascoyne, not so different on this large scale, except if you're looking particularly in the bed of the river. Here's the wet season, here's the dry season mosaic. Here's just that area around Carnarvon that I mentioned, where we have a lot of fruit plantations and berries, orchards, etc. Here's the wet season, here's the dry season. Very different pattern depending on the seasonality of the particular crop that they're growing. And here we have Oyster Harbour around Albany. Here's the wet season and here's the dry season. A very different north-south trend there, which is dependent on whether or not it's this intensive agriculture versus that native vegetation. Okay, so how do we link what we're seeing in terms of the land water usage within the estuary catchment with the estuary water quality? How do we link these two parameters? So here's just some qualitative approaches that we used initially in this project. So firstly, let's look at the numbers down the bottom there of land use, land cover that relate to natural environments. So that's just outlined here for you, and you can see that there's some anomalies that pop up in the hundreds here in the Gascoyne in particular that are quite different. So we have in general, you can see that natural environments are clearly identified by their evapotranspiration trends. They have much lower dry season evapotranspiration because they are largely rainfall dependent and they are native plants, so they're very smart with their water usage. So when there's less water available, they drop back, and then when more water is available in the wet season, then they green up and their canopy and their vegetation density extends. So you can clearly see them from and identify them from their evapotranspiration trends. They have a very strong seasonal response and they're receiving little inputs in excess of rainfall, except for some of these ones that you can notice here, particularly in the Gascoyne, and that allows us to identify areas that may receive additional inputs beyond rainfall, such as vegetation that may be tapping into groundwater, for example. So how do we link this to water quality? Well, one hypothesis might be that because this is vegetation that really dries back, really dries off, really falls back when there's less rainfall, we see a massive ET decrease and to a very low level, meaning that there's very minimal vegetation coverage in the dry season. That tells us that there's going to be greater soil mobility, and in the rainy season, potential transport of soil into the waterways. That tells us that we could anticipate that there might be an observable increase in total suspended matter content. So let's look. Let's see if in the wet season, do we see this spike in total suspended matter content that could be linked to this drop back in vegetation coverage because of this rainfall dependent vegetation that dominates the Fitzroy and Gascoyne catchment areas in particular. So if we look at the Fitzroy, this is our total suspended matter for the area with the outflow of the Fitzroy River in the wet season and the dry season, and what we see is consistent with our expectation. Typically a 50% higher total suspended matter content in the wet season compared to the dry throughout the estuary and King Sound. So that's consistent with our prediction of higher soil mobility, consistent with this land surface water usage indicating significant vegetation coverage loss. But you have to be so careful, particularly with interpreting the Fitzroy, because the Fitzroy has these massive tidal patterns, and so there's a big caveat really on our interpretations of these products when it comes to the Fitzroy region in particular. So let's look now at the Gascoyne. Here we have the mouth of the Gascoyne River estuary discharging into the ocean there. So if you look particularly at where it discharges and flows northwards, upwards into the reef areas, what we see is quite complex. That nearshore reef area where it discharges and flows northwards, it is considerably higher in the wet season than the dry season, which is consistent with our predictions about the soil mobility. But if you just look further outwards into the deeper ocean, then that's actually higher in the dry season. So this may be complicated by the lack of flow of the Gascoyne in the dry season. It's this upside down, unusual river. So this is a somewhat complex system as well. So it is not clear to link the two in that area. So what about these irrigated agricultural environments that dominate the Oyster Harbour catchment and also a portion of the Gascoyne catchment around Carnarvon? So that's these higher numbers that you see here. in the 400s. And you can tell immediately that they're very distinct in their evapotranspiration trends. They have substantially elevated dry season water use, elevated wet season water use compared to natural vegetation environments. That tells us they're getting significant additional water inputs all throughout the year. We can also look at their dry-wet ratio as well as being a diagnostic feature, but I won't go into that. So, what prediction can we make? Well, we could predict that they're receiving potentially chemical inputs throughout the year in the form of fertilizer because of this persistently high evapotranspiration, persistently high vegetation coverage. So we would expect to potentially see runoff inputs of chlorophyll, of fertilizer, leading to a potential spike in chlorophyll A in the estuary during the wet season. So, do we see any evidence for that? In the Fitzroy, yes, we do. The Fitzroy has very low levels of chlorophyll A, which is what you would expect, it being our pristine site. But it is consistent with getting a spike after high rainfall events, which is what you would expect. So there we have our wet season on the left and our dry season on the right, and you get a spike in chlorophyll A. We also see the same in the Gascoyne, which is consistent with our predictions of increased nutrients as inputs into the estuary, potentially linked to these high rainfall events and this bi-seasonally high land surface water usage of vegetation, particularly around Carnarvon. So that's what we see here in the wet season and the dry season at the Gascoyne. So I haven't spoken much about Oyster Harbour because it is a bit more of a complex estuary and a difficult one really for us to do without some ground truthing at the site because it is such a shallow estuary. So there was caution in our interpretation. So what we see is some really interesting features that I'll show you here, particularly in the discharge of the Calgon River into the estuary here in the wet season and the dry on the right, and here in the flushing of the Emu of the ocean, between the estuary through Emu Point here. The key messages from this are really that evapotranspiration can be used to identify the potential sources of land use and land cover that are leading to the inputs of chemical and sediment into the estuarine environments and inform the land water management for these areas. So we've contributed some data products that the Department of Water and Environmental Regulation can use to assist with decision making around future estuarine management and their ongoing monitoring programs at these sites. And although at the moment we've been talking about quantitative relationships, what we need to do is to establish these to quantitative. We've been talking about qualitative. We want to leverage these into quantitative relationships. And what we need to do this is really have a dedicated observing system where we're measuring each key variable in situ. So sap flow sensors to measure evapotranspiration and to validate and if needed calibrate our satellite derived products, and marine sensors to measure our TSM and our CHL products as well. And then once ground truth, these products can be used to provide a baseline to assess the impacts of any future monitoring and management interventions that we look at doing to try and improve and minimise the human impact on these precious estuarine environments. So thank you very much for listening to me today, and if you'd like to chat to me about any of my other work in using evapotranspiration and the applications of it, particularly to vegetation monitoring and sustainable water management and to bushfire severity risk, please do get in touch with me. Thank you very much for having me. Bye.
Well, I'm going to take you underground because everything seems to have been above ground so far. And I want to point out to you that our knowledge of that is skin deep. This is really Wayne Meyer is the senior author on this, and I'm presenting it on his behalf with quite a bit of data from Sally Thompson. If you look at this on the right-hand side, the left-hand side there, you'll see that this is a global data set of soil carbon, and you can see that the vast majority of it is in the top 30 centimetres. We have very little understanding about what's going down underground. That doesn't matter too much if you look at the centre graph there, where your bedrock is at 50 centimetres, but if you're interested in carbon stock and carbon nature of the carbon, then if you look at the Hawkesbury profile on the right, you can see that if you're only interested in the top 30 or measuring the top 30 centimetres, you're going to be way out because there's quite a lot of carbon further down the profile, and that's only to 1 metre, and the profile is much deeper than that. pick your system. Really, our understanding really is much poorer with soil depth, and this example here is on nitrogen accumulation, and you can see that this is from fertilisation. You can see that the organic nitrogen is down at 20 to 50 and 50 to 100 and probably even lower. So if your sampling and understanding is only at the very surface, you're going to be way out. This is the number of soil nitrogen measurements. You can see there that there's a very much larger number of observations at 0 to 30, and you can see that decreases quite markedly once you get down to a metre. So this led us to, in 2001, a group of scientists in the United States decided to conceive of the critical zone. And this is where life really depends on this, from the upper atmosphere, the low atmosphere, the organisms, the biosphere, then going underground to the regolith, soil, and down into the groundwater sitting on the bedrock. They conceive this as the critical zone, Earth's living skin, sitting between two very big physical systems, the atmosphere above and the lithology below. And they set up a series of critical zone observatories in order to study this particular process. And it's been brought in over the last 20 years or so by quite a large number of groups, and there's around about 45 critical zone observatories which attempt to measure that system from the near surface atmosphere through the vegetation, the biosphere on the surface, through the regolith and the soils, down into the groundwater. But that graph, that map there shows you that the vast majority of those sites are in the northern hemisphere, and there's virtually nothing in the southern hemisphere, and yet the pedology, the geology, the lithology is very different. The northern hemisphere, sort of just arm waving here a bit, was largely swept clean by the ice sheets 5,000 years ago, so they're much younger soils and very often shallower, quite shallow, relative to what we have in the southern hemisphere, which escaped those ice sheets, and therefore we have much higher weathered, larger weathered soils and much deeper. Their chemistry, mineralogy, this is arm waving, and of course is quite different in many cases. So Australia, Southern Africa, and South America there. So what does it take to understand the critical zone? Well, I'm not going to focus too much on the above ground because that's been covered by a number of speakers so far. But what I want to point out to you is with the critical zone observatories, the distinguishing feature of these is that we're going underground, in which we have groundwater wells down to the groundwater and monitoring that, its chemistry and flux. But in addition to that, we have these vadose monitoring systems in which we look at the chemistry and the gases that are in the unsaturated zone. So we're able to monitor that in real time, thanks to some innovations from a group called Sensoils in Israel that have set these up in 3 of our locations already, and we're about to extend that. So we're linking very closely with OSCO, with the geophysics, and also obviously with TERN, and we have to thank very much our OSCO team, which is a 5 universities to Beryl Morris for incorporating us, or trying to, into the TERN system, and also her encouragement. So what with ARC funding, we've got an infrastructure grant from them, and it's been pretty frustrated, I must say, from as a result of COVID, but we've managed to set up these critical zone observatories in 5 locations, and we should be able to finish the job off towards the end of this year, and we're in 5 locations. In Pingelly, over here in southwest Western Australia, in Calpram Station, in the River Murray, Mallee area, north of the River Murray, at Wellington here, which is run by the University of New South Wales, and in the Fletcherview field station here, and we hope that, and Moreton Bay, I should have realised, that's one that I'm involved with in the campus of the University of Sunshine Coast. We realise that it's not comprehensive, if you like, of the regolith and of the Australian continent, but nevertheless, there's a good deal of climatic variability, of course, and vegetation variability there, but the key thing that is important here is that they're all deeply weathered profiles. Or in the case of Calpram here in the River Murray, there are some young soils on top, but nevertheless the parent materials of those soils have been gone through very large number of erosion cycles. This is a contribution to the global system because there's, as I showed you on that map, there's very little, if anything, in the southern hemisphere, and yet that geology, lithology, pedology is very quite different compared to the northern hemisphere. So this is helping to make a true global critical zone observation system. There have been a number of fundamental hypotheses developed, which is just being discussed at the moment in the United States based on the work over the last 20 years since these were established. I'm not going to go into any of these in great detail, but simply to mention that these are hypotheses generated from the data that's come from those sites almost exclusively in the Northern Hemisphere. And if you look at the bottom 2 there, for example, you can see surface reaction fronts create subsurface flow paths. So it's this deep process through the regolith, the soil, deep soils, that is quite critical to understanding the functioning of that critical zone from the near surface atmosphere through to the bedrock below. And humans change the critical zone from a zone of transformation to one of transportation. So these are going to be worked on in various ways and being discussed now as to how we might be able to put more research into those particular hypotheses. Now I want to switch you to Calpram, which is a TERN supersite, and give you some data that has been generated by Wayne Meyer, my colleague. And what you see here in black on that graph there is the rainfall, and you can see that it's quite spiky. There's a great deal of variation seasonally, but the key point that we've noticed there, or measured there, is that the rainfall has gone from 250 millimetres per year, the long-term average, to 180 millimetres since 2017. Now, a simple water balance model here, where you just look at evapotranspiration and rainfall, very simple, nevertheless gives you some idea of drainage. And so in red there is the evapotranspiration, the rainfall indicated there in black, and then when if the evapotranspiration is greater than the rainfall, then you would expect to see through the root zone water rising, and then if it's the other way around, rainfall greater than evapotranspiration, we'd start to see drainage through that root zone. And you can see that on this plot here that there is a great deal of, well, there's limited drainage here compared to previous 2017. There's been quite a change, as this plot here, my colleague Wayne Meyer and some of his assistants has generated. So this is the total volumetric water content in the top 2.5 metres of the soil profile as measured by neutron moisture meter, and you can see here that's what you see on the y-axis there, the total water content to 2.5 metres, and the date from 2014 to 2022, and you can see that from around 2017 there's been a marked change, and we're down to the lower limit of the plant available water at that point. So it would indicate that there has been this persistent drying since 2017. And what are the consequences of that? Well, this system is under stress, and you can see that this is leaf area index, and there's been strong regrowth here from 2014 following a fire, and I'll show you some photographs of that shortly. But then there's been this soil drying that I've indicated from that previous plot, showing that there is a severe drying, and also it's reflected in the leaf area index. And this is showing that the next slide will show you that this system is really under stress. And you can see these grubs came in just recently. It's a photograph that Wayne sent me, showing that this was only in a matter of 3 days. It really impacted the vegetation greatly. You can see the browning off of the vegetation in the foreground and also in the background. So it's over a very extensive area. So here's the question. Is this a relatively short-term effect, or is this evidence of a new climate or ecosystem regime? Is this a tipping point, in other words? So here's the questions we have at Calpram Critical Zone Observatory. We've got to the stage at this point of, it's been a long-term supersite since 2012, everything pretty much above ground apart from down 2.5 metres where we had the soil moisture measurements, but now we've put the groundwater wells into 45 metres, and which is where the groundwater is, and we've also got about to put in the vegetation monitoring system. Where is the upflow of the water coming from for this vegetation if there's been such a marked amount of drying out? What are the consequences for the interaction with the regional groundwater? So we've put up a few research questions here, which I won't detail now. I'll finish off with these slides to show you how stressed this system is and our feeling that this is under a tipping point. We're at a tipping point at this system. 2012, there's the native vegetation. Have a good look at the understory. There was the fire in the summer 2013-2014. There's the regeneration in 2019 in that part there. Here's 2020. You can see some regeneration. So the water's coming from somewhere. We don't know where. The vegetation monitoring system in particular should be able to give us an idea of where that water's coming from and how much is left, because have a look at the understory. So there's some really serious issues here, which we hope the Critical Zone Observatory around the country will add, particularly to the TERN, but we're going to be also linking quite closely with AUSCOPE, the geoscience agency, as well. Thank you.
Good afternoon, everyone. My name is Ben Umali. So just here to present to you an update of work that we've been privileged to undertake with like-minded people at TERN. So this is a piece of work we began to undertake this year at Fletcherview Supersite, which is found in the northeast corner of Australia. Joe already mentioned to you earlier this morning about the site, but generally this is dominated by Reed River box and native shrub Carissa bush, and it drains to the Burdekin River and is a major tributary to the Great Barrier Reef. This slide gives you a rundown of what's currently on site, which was also mentioned earlier by Joe. But again, also critical here are the people that tirelessly conduct research, including data collection, monitoring, and site maintenance. All right, so why plant water status, you might ask. At ICT, we believe that plant is the ultimate transducer. Plants are good at integrating what the environment is throwing at the plant. Being able to measure the response in situ is gold. So the ability to measure tree water use and plant water status are two important parameters, sometimes often neglected. We always see weather stations, soil moisture probes, proximal sensors like spectral cameras, but in our disposal are sensors that can track high temporal resolution SAF flow sensors and stem water potential sensors. So these instruments can measure species response to its environment and over time insights into how plants may be impacted by changing climate or even by other plant species in the community as a result of, say, disturbance and introduction of invasive species. So and in the last February's iLeaps Ausflux meeting in Auckland, we also met like-minded people like Joe and George Verba and thought, hey, why don't we start looking at these two parameters and show how this can add value into ecosystem monitoring. So and Joe is keen in looking at evapotranspiration partitioning and agrees that plant-water relation measurements she'd be able to do that objective. So SAF flow sensors. Here's what we've installed in the field. It's a SAF flow sensor. There's in the flesh out in the exhibition area. So it's an Australian product. Probably not many of you may know. It's been used by some of you, but it's been used across the world by many scientists. It's been published probably 400 times. It's the only standalone product available in the market, well liked by eco-physiologists because of its robustness and how easy it is to deploy out in the field. The sleek design means scientists don't have to contend with wires, which was a characteristic of many SAF flow sensors in the past, and the heavy post-processing of data. So it all happens inside the instrument. So we instrumented three tree species, and we deployed a total of, I think, over a dozen sensors across 50 by 50 meter plot within the edicovariance flux tower footprint. So the next sensor we installed is a couple of stem psychrometers. If anyone has ever measured pre-dawn and midday water potential in this room, will realize that this instrument, you'll never have to wake up in the middle of the night or stay under the sun and carry pressure chambers in the middle of the field to take water potential measurements. So these instruments allow you to capture diurnal and long-term patterns of plant water potential. And we instrumented 2 species in Fletcherview, but we hope TERN community or TERN scientists will start to use this more and more in the future. It's been cited in hundreds of publications, so you don't have to worry about the integrity of the instrument. And it has been used to look at silent vulnerability and overall plant response to drought. In the installation process, Joe took care that there's good representation of the measurement. We assisted in the gear installation, and so the gears have got internal batteries in them. But for prolonged measurements, all units or all sensors were connected to an individual 20-watt solar panel. The installation sites within the trees were prepared properly using appropriate protocols. So ICT have extensive or well-documented protocols in the installation. Sensors and loggers were insulated to minimize impact of abrupt temperature gradient. So what have we seen so far? Right off the bat, a few weeks after the sensors were installed, one apparent thing is the variability in water consumption of tree species that we've measured. Rimophila mitchellii used twice as much as the Eucalyptus brownii, which is in turn using 4 to 6 times as much as the Carissa bush. Well, there's really nothing surprising there. But if the name of the game is understanding how much each tree is contributing to the total water consumption of the community, and thereby water balance, then this certainly puts you in a better position to work that out. Eucalyptus mitchellii may be consuming more, and now that you have data on individual water use, and if you know how much of these species occupy the landscape, one can extrapolate total water use of this particular species in your landscape. Because the instrument was set out to log 10-minute measurement interval, I can zoom in on a short time scale of my measurement, or your measurement. Here I can interrogate daytime and nighttime patterns of water use. This allows me to look at individual species patterns at the time scale and compare their behavior. Questions like, why does Eucalyptus brownii exhibit what looks like a basin during the middle of the day, while others seem to follow sun-induced transpiration patterns may begin to form? Okay, most of you might know the answer to that question already, but I'm sure you will appreciate the granularity of this data and form deeper questions related to the eco-physiological behavior of these species. You can almost see what is happening in the moisture regime of the surface soil. One will notice the gradual decay in the water use of Carissa, the orange line. That's over 9-day time course. The river box and fall sandalwood, presumably due to their deeper and extensive root system, are able to access greater soil moisture pool, and so have not declined over the same time period. Now, what about water potential? Here I overlaid 5-day water potential measurement in the secondary y-axis and the sap flow of Eucalyptus mitchellii. The water potential data is represented here by brown line. So please note that this is increasingly more negative. Observe how negative it can get as soon as the sun comes out. -7 megapascal just around 10 a.m. in the morning. Stem water potential is related to the energy that it exerts to pull water from the ground. So number of observations can be observed here. You see nighttime transpiration there, whether that's water refilling or nighttime transpiration or nocturnal transpiration is something that we should be asking. There's really a lot to learn from the different plant species in Fletcherview Supersite using this instrumentation. There's certainly a lot more to learn across the different TERN sites across the country. It will be exciting. What we can learn from measuring sap flow and psychrometer water potential measurements across the season and across many years for long-term ecological investigations. Thank you very much.
Thank you for having me here. Thanks. So I'm the guy who measured pre-dawn water potential for 2 months. Yeah, thank you. So today, the main purpose of the talk is just to improve the transparency between the biological processes, the physiological processes. We think, or we using experiment has tested that it's important and to increase its incorporation for the modelers to improve the model performance. So this is a slide really generated by Belinda. She's here. So generally, we have hundreds, thousands of drought experiments at different setup in glasshouse, manipulated, controlled, open tap chambers, or in field. And also for modelers, so they are good at simplifying things, so they can simplify a whole ecosystem or an entire continent as a single piece of leaf, and then attribute the values for each K parameters they think are important to that piece of leaf, and then do evaluation of the model performance. So if we can improve the transparency between, basically for experimentalist or experiment, we understand what will be the key processes, ideally in a quantitative way, for modelers to uptake. And also for modelers, if we can understand what are the key processes, physiologically, biologically, and what is the good way to quantify those processes in our models, that would be nice. So in that case, we may need meta-analysis, we need to understand the processes better, and we need to understand how to quantify the processes. So here's just a quick example. This is a picture generated by Martin. So here are the large models, different of them, and beta is a function, a coefficient the model will use to represent how severe the water stress it is. So basically it's a coefficient, the number is from 0 to 1. So you say the performance, the representation of this beta function in the different models is different in terms of when would the water stress start. It could be starting very early, it could be starting very late, and how quick the water stress will affect the function in that model. So some of them have a very sharp response and then shutting down very quickly, and some of them will adopt a very gradual decline of the photosynthesis function in their model. So the key processes we identified as important. So basically, take the chloroplast as a factory to produce carbohydrates. So we need the raw materials, that's the CO2, the carbohydrate, to be delivered to that manufacturer factory. So on the logistic of the transportation, there is one door here, that's the stomata. So that will affect the diffusion of CO2 from atmosphere to the intercellular spaces. So that's stomata conductance as one limitation here. Then when the CO2 enters into the intercellular space, it needs to arrive at the factory. So on this logistics, on the road, there is limitation by the mesosphere conductance, so mesosphere conductance. And when it arrives, how efficient this factory in terms of the production efficiency. So it's limited by K biochemical parameters. So this is VC max, so that's the maximum Rubisco activity. Jmax, that's the maximum electron transport rate. So basically, we can design controlled water stress experiments, and we can understand how would each process get involved, and then we can quantify their response function. If we can quantify them, then we have a very transparent way for these quantitative functions to be incorporated into models. And with our models, we can compare our field measurements on the fluxes set up in field, and then compare if our new incorporation of those biological processes will improve the performance of the models. Yeah, so here basically currently the some models, they just use the ratio between stomata conductance and photosynthesis rate as the beta, the coefficient of representation of water stress in their model. Some models considered the Vcmax and Jmax, and yeah, we just need to use experiments to inform what should be incorporated and how to incorporate in a quantitative way. So here are the key questions, yeah, in terms of that. So basically, we set up two sets of experiments. So one in Australia, one in University of Barcelona in Spain. So for the Australian one, we collected eucalyptus species from mesic hydroclimates and also xeric hydroclimates. So this one is like a super performer in terms of water adaptation and water acclimation. I will show you data later. So for the Spanish experiment, experiment in Spain, so we collect the Quercus oak species. So from the xeric, semi-xeric array, and also the mesic array. So, and also two species that has been well known. They are very water sensitive. So we put them independently in a glasshouse experiment and manipulate the water stress. So basically, we water them from like 100% field capacity, fully water them, and then stop the watering, just let them to drying down until the day of stomata closure. So during the whole process, so for this species, that's about 48 days. So each day I will measure the pre-dawn water potential, the photosynthesis, and just to quantify like the Vcmax, Jmax in terms of the photosynthesis response curve in terms of the CO2 response. So here is just a quick show on the response curve fitted from raw data. So here's stomata sensitivity, mesophyll conductance, the maximum Rubisco activity, and maximum electron transport rate. So this curve, this is the species, this one is, sorry, this one is not here, but this one. So generally the species from the Western Australia, the xeric hydroclimate, is like a super performer in terms of the starting point. It started to show the stress symptoms at a quite later stage compared to the riverside species. It just, they cannot sustain for 2 or 3 days. So for the xeric species from Western Australia, so it can still keep its, let's say, its Vcmax to its like to half reduction of the original value, even after 48 days. So that's, yeah, quite a good performance. So by collecting the data and by fitting the data with the best response function, so for the diffusion, that's stomata conductance and mesophyll conductance. For diffusion, we fitted the data with an exponential function. For Vcmax and Jmax, we fitted the data with a logistic function. So with this function, so we can quantify what is the value of G1 start, the starting point, and what is the value of the sensitivity of the decline rate of each curve, B1, for each species. And also we can quantify the K parameters for the response of Vcmax and Jmax. So by doing so, we can incorporate these K quantitative traits, incorporate their value, bring these parameters and the values of differential species from xeric versus mesic sites into model and try to test the performance of the model. So here is just a quick show on how we did it. So the very drought-resistant species, the value of the parameter, and also very sensitive ones. So yeah, so here just a quick show that, yeah, so it did bring the new insight to the model performance. So the further work by Martin and Belinda and Cornelius so identified, so our approach is necessary to ensure the model to have a realistic representation of the flex tower observations in field. So here just a quick show. We have several presenters mentioned about acclimation. So here is one experimental evidence that acclimation can really happen. So basically, we have extended the drought experiment just from 2 months to 4 months. We do the same thing of, like after 4 months of drought treatment, we did the same drying down process from 100% field capacity to stomata closure. And we found like those change of the normal parameters that we generally measured by most drought experiments. But what we found is the differential acclimation capability. So basically, the species from the Western Australia xeric area, after 4 months of drought training, its VC max response kind of being more resistant to water stress. So it kind of like shows a signal of acclimation after 4 months. So in terms of that, you say the other species, the other 2 gum tree species from the mesic environment, they showed no acclimation in terms of that. So this is kind of like a significant thing. Yeah. So I think generally I will just basically stop about here. So what we want to do is to incorporate the above ground incorporation of experiment into a modeling. How can that be transferred or connected with above ground? So like David just showed. So this is super important. So a potential way could be, so because we have spectrums of leaf economic traits for species from xeric to mesic climate origins, and also there are evidences showing the root traits can also do like in a similar way, but there are many other papers disagree. So, and there could be the whole plant above ground and below ground can be integrated in this approach. So if we have spectrum, we can use spectrum, put this spectrum into modeling. So that's easy. So this is just, yeah, okay. I think I will stop here. So we just, yeah, thank you, thanks.
So thanks everyone for really interesting presentations. We've got 10 minutes until afternoon tea, and I've got a few questions to start with. I've got a couple for Maree, and I'm going to try and roll them into one. I guess the trade-off between economic returns and land degradation, that constant one, in the frame of maximising profits. I guess going to the regenerative agriculture, or the avoiding the boom and bust, if you like, is there how do we capture, can we capture that somehow in an economic model as a risk approach that dampens the value of the boom and reduces the heartache of the bust?
Yes, that's a difficult question. We need good data to understand all of these effects and to model them. The way we do our analysis is over a 30-year time period at least, so it's a long-term perspective. And the key metrics are net present value, so profitability, and you can determine an annualised change in profit per year, but also the peak deficit. The financial risk measures a peak deficit and days to payback. So because of the way economic analyses work and the time value of money, it means that near-term profits are valued more highly. So, this is why the sequence of these events in time is important too, whether you start during a drought or whether you start at a series of good years. So the modelling needs to take into account all of these factors, as well as the peaks and the troughs and the dampening. So it's quite a difficult, it's quite a challenge, and the bigger and more complex the model that tries to incorporate climate variability, the more difficult it is to do the economics properly, so to do a marginal economic analysis of change, which really gives the information that influences producer decision making. So that's why there's a real lack of studies that combine all of that, because it's not easy to do. So that's a long-winded answer, and I hope that's answered the question somewhat.
Yeah, well, I think it does highlight the complexities of human decisions we have to make over long time frames that are difficult to measure to capture the change over that time frame. So thank you. I've got a whole lot of orders here for you, Will, but no questions yet. I'll get there. I've got one. To Dave, with the Critical Zone Observatory, I've got a question from about the consideration of extending the observatories, if we are looking at extending the observatories into managed landscapes, so that up in the Brigalow country, for example, where we've got a long history of land management and the influence of land management in, the role of the critical zone in terms of management outcomes in these managed landscapes.
The answer to that is yes, we'd like to see this extended. We have captured a broad range of environmental parameters there in climate, vegetation, soils, with the 5 that we've put in. But that's just been done with a $2.2 million ARC grant, with some monies coming in from the universities as co-contribution. It's an infrastructure grant, and our funding runs out on December the 31st. So we hope we don't have a white elephant.
Yeah.
And for there, I have to thank the team very much, to Beryl and Tim Rawlings of OSCO, for embracing the Critical Zone Observatory concept and trying to incorporate it into their programs in an integrated way. And of course, 3 of the sites that we have of the 5 are in fact TERN supersites. There are 3 sites around the country which we might call satellite sites, one at Gin Gin, Springwood, in the up in the heritage zone in out of the Gold Coast, north of the Gold Coast in the ranges, and we wanted to have one in Kimber, which then extends the thing away, the concept away from, if you like, ecosystem work into the radioactive site, because that's going to be, well, we're not sure now after the court case this week, or last week I should say, where the challenge was put out, but Australia's medium waste repository is put on hold now, even though the regulator has got 200 hectares of land there at Kimber. So we see the importance of extending it, but our real key point is to try and get operational funding, and we're working on that with the help of Beryl and Tim. Yeah.
So I'm assuming it's like the rest of TERN and There's just as mere smell of an oily rag is enough to run the site for a decade. How are we going for time? 5 minutes? Okay, good. If there are any other questions in the audience, grab my attention. It's really hard to see from up here, down here, up there. You're all very dark. But I had one which I think might be related to two things: our last presentation on the glasshouse experiments and our Fletcherview, the ICT instrumentation. Can we bring those technologies together to address some of these physiological questions using field data, particularly of older systems, more ecosystems through that are stable and not a seedling? So just to compare the results of the two. So I'll let you both have a go at that if you don't mind.
So I just don't understand 100% what my colleague from Western Sydney University discussed earlier. It's way above my my knowledge, but I believe if we're talking about more data collection and how that measurement relates to our measurement would be a good I guess synergy. So yes, again, what our instruments can do is that it can give you in situ measurements out in the field for different species, and validating the models that you guys wanted to validate, that you've developed in your labs, in your computer simulations. Then with instrumentations, field instrumentations, then that will give you more, I guess, confidence about the model that you churn out down the track.
Thank you. I think the technology is, in terms of, let's say, sensors or the way of doing the measurements, the way of collecting data, I think that's a lot problem. I think the key thing is to bridging, incorporating, translating. So when we're saying about that, it would be between glasshouse experimentalists to people doing field measurements, and also all these set of measurements, how would that, the communication between modelers or modeling work. And also that will involve a lot of infrastructure, sensors, technology. It could be radar, LiDAR, it could be different sensors. And also, I would also like to say, in terms of for ecosystem, tomorrow we will have more talks in terms of the carbon neutral that will involve huge agri-system. So for agri-system, that will involve a lot of interest from the industry, from the commercial world. So how would that be communicated, translated between, right? So that water technology, everything, I think WEIR has a great example in terms of how to simplify the fundamental knowledge, fundamental philosophy, fundamental science to be translated to the applied world. That's very important. I think we need to open that transparency a bit more. Yeah, I encourage every people to think about that. Thank you.
Thanks. Thanks, everyone. And a big round of applause for our speakers this afternoon. Thank you, Beryl. I better give that to Mark.
Thank you, everyone. And you've been so really great at this audience bit that we've just done a little vote, decided to give you a little extra time for afternoon tea. We're now asking you to be back here by 4:45. So you have half an hour, 25 minutes. We didn't give you that much. So please enjoy a quick break. Thank you.